A single tree once provided more food than an entire acre of farmland. For thousands of years, the American chestnut, Castanea dentata, dominated the eastern forests, comprising nearly 1 in 4 trees across Appalachia. A 1904 survey by the U.S. Bureau of Forestry documented vast ridge systems where chestnut canopy exceeded 30% of total forest cover, with individual trees reaching 30 meters tall and producing hundreds of kilograms of nuts annually. Indigenous nations from the Cherokee to the Iroquois built seasonal economies around its harvest, storing, trading, and eating chestnuts as a primary caloric staple before European contact. This was not a background plant. It was the keystone species of an entire civilization's food system. Settlers called it the breadnut tree. Railroads were built from its rot-resistant timber. Children filled their aprons with its sweet nuts every autumn. Furniture, fence posts, tannin for leather. One tree served a dozen industries. No single species has ever been so completely woven into the economic and nutritional fabric of a continent. And no species has ever been so completely erased. What killed 4 billion trees in 40 years was not a storm, not an axe, not a war. It arrived quietly, on one uninspected shipment of nursery stock. Understanding what was lost requires understanding what the chestnut actually was, biologically, structurally, ecologically. Castanea dentata was not simply a large tree. It was a mast producer of extraordinary consistency. Unlike oak, which produces acorns in unpredictable cycles, the American chestnut fruited heavily every single year. That reliability was rare in the forest economy. Wildlife, bear, deer, turkey, passenger pigeon, depended on the fall mast as winter preparation fuel. Human communities scheduled their calendars around the chestnut harvest. The nuts contained roughly 45% carbohydrates and were one of the only tree nuts nearly free of fat, making them nutritionally closer to grain than to walnut or hazelnut. They could be dried, ground into flour, boiled into porridge, or eaten raw directly from the burr. European settlers recognized the parallel immediately. They called it the American equivalent of the European chestnut. But Castanea dentata was faster growing, taller, and more cold-tolerant than its European relatives. Its timber was straighter-grained, lighter, and extraordinarily resistant to decay. A chestnut fence post, driven into wet Appalachian soil, could outlast the farmer who planted it. The tree gave everything: food, shelter, income, and ecological stability. The forest without it would become structurally different. No one knew how different until it was gone. The year was 1904. The Bronx Zoo in New York imported a collection of Asian chestnut trees, Castanea crenata, for ornamental planting on its grounds. No phytosanitary inspection existed. No protocol screened incoming nursery stock for fungal contamination. Hidden within the bark of those ornamental imports was Cryphonectria parasitica, a microscopic ascomycete fungus native to East Asia. Asian chestnuts had evolved alongside this pathogen for millennia. They carried the infection without dying, a biological tolerance built over thousands of generations. The American chestnut had never encountered it. Its immune architecture had no answer for what was coming. By 1905, American chestnut trees near the Bronx Zoo began showing orange cankers on their bark. By 1910, the blight had reached Pennsylvania. By 1920, it crossed the Appalachian ridgelines like a wave. The fungus traveled on wind, on birds, on logging equipment, on boots. It entered wounds in the bark, colonized the cambium layer, and strangled the tree's vascular system from the inside. Scientists watched, largely helpless, as the most dominant forest tree on the eastern seaboard began to die. 4 billion trees in 40 years. the largest ecological catastrophe in recorded American botanical history. Cryphonectria parasitica does not simply infect, it engineers death. The fungus releases oxalic acid as it colonizes bark tissue. That acid lowers the local pH so dramatically that the tree's own cellular defense enzymes are deactivated. The cambium, the living layer responsible for nutrient transport, is destroyed in a ring around the trunk. Botanists call this process girdling. Once girdling is complete, everything above the canker dies. But here is the biological paradox that defined the next century of chestnut research. The roots survive. Below the soil line, the American chestnut root system remains alive, sometimes for decades after the aerial portion dies. Shoots emerge from the root collar, grow several meters, then are reinfected and die back again. The tree exists in a kind of suspended biological grief, alive underground, dying repeatedly above. Researchers at Cornell University documented root systems from pre-blight trees still sending up sprouts in the 1990s, nearly 90 years after the original infection. The forest floor remembers what the canopy forgot. This persistence became the foundation of every restoration attempt that followed. The American chestnut did not go extinct. It went underground, waiting. When 4 billion trees disappeared from the eastern forest, the ecological consequences were not immediately understood. Ecologists in the mid-20th century had no baseline for such a loss. Later research revealed the depth of the damage. A 2013 study published in Forest Ecology and Management documented that chestnut's disappearance fundamentally restructured understory light dynamics, soil chemistry, and mast availability across 12 states. Wildlife populations that depended on reliable fall mast, particularly turkey and bear, shifted range and behavior patterns. Oak and red maple expanded into the ecological void, but neither replicated the chestnut's caloric density or fruiting consistency. The passenger pigeon, already under pressure from hunting, lost a critical winter food source. Some ecologists argue the chestnut's collapse accelerated that extinction. Appalachian farming communities lost an annual source of income. Rural families had sold chestnuts by the bushel at market every autumn. That income stream vanished within a single generation. The timber industry lost billions of board feet of the most commercially valuable hardwood in the region. Tannin extraction, a major industrial use of chestnut bark, collapsed entirely. One tree's disappearance rewrote the economy and ecology of an entire mountain range. No replacement was ever found. No species stepped fully into the void. In 1983, a small group of plant breeders and conservationists founded the American Chestnut Foundation. Their goal was precise: restore Castanea dentata to the eastern forest using backcross breeding. The method involved crossing American chestnuts with blight-resistant Chinese chestnuts, then repeatedly back-crossing the offspring with pure American stock. Each generation recovered more of the American tree's genetics, while retaining the blight resistance gene from China. After 6 generations, the resulting trees were approximately 94% genetically American chestnut. Field trials began in the 1990s across multiple states. The trees grew tall and straight, with the timber profile of the original. They flowered. They produced nuts. But the blight resistance proved inconsistent under real forest conditions. A parallel approach emerged from the State University of New York: genetic engineering. Researchers introduced a single gene from wheat, encoding oxalate oxidase, that neutralizes the oxalic acid the fungus deploys. Transgenic trees in controlled trials showed dramatically reduced canker formation. A 2020 paper in the journal Plants confirmed the mechanism's effectiveness without apparent impact on surrounding soil ecology or mycorrhizal relationships. 2 paths toward the same destination. Both carry the weight of 4 billion silenced trees. In isolated mountain hollows across Virginia, North Carolina, and Tennessee, oral traditions preserved what the scientific record almost missed. Elderly farmers interviewed by ethnobotanists in the 1970s described the chestnut harvest in sensory detail. The sound of burrs splitting on frost-hard ground, the smell of roasting nuts in iron skillets. One documented account from a 1972 Foxfire interview described a single tree producing enough nuts to feed a family through October and sell bushels at market through November. These accounts became primary sources for researchers reconstructing the tree's historical ecological role. The memory was not abstract. It was caloric. It was economic. It was architectural. Entire barns and cabins stood on chestnut beams that outlasted 3 human generations. The tannins in chestnut bark were so potent that leather tanned with chestnut extract was documented to outlast oak-tanned leather by years. That knowledge nearly vanished with the tree itself. European imports eventually supplied the tannin industry, but at economic and ecological cost. The loss of the chestnut was also the loss of a specific kind of self-sufficiency. One tree had provided food, building material, income, and industrial input simultaneously. No crop domesticated since has replicated that combination. The Appalachian mountains still grow chestnut sprouts from ancient roots. The land has not forgotten what it was capable of producing. The American chestnut is not a ghost. It is an interrupted story. Thousands of root systems across the eastern United States still send up new growth each spring. Restoration orchards from Maine to Georgia now hold back crossbred trees in various stages of evaluation. Seed banks preserve genetic material from pre-blight specimens. University programs train the next generation of chestnut breeders. The transgenic trees await regulatory approval, a process that has taken decades. The debate between genetic engineering and traditional breeding reflects a deeper question about what restoration actually means. Is a tree restored when it looks right? When it grows in the right place? When it carries the right genetics? Or when it feeds the forest the way it once did? The American Chestnut Foundation estimates that restoration, if successful, would eventually return billions of trees to eastern forests. That would represent the largest deliberate reintroduction of a lost species in botanical history. 4 billion trees were lost in 40 years. The question now is not whether the species can survive. The question is whether the system that destroyed it has changed enough to let it return.